This presentation is talk about some of the challenges that we're confronted with microbial identification as well as antimicrobial susceptibility testing. If we start with microbial identification, it's really interesting. As I said, that when I was at the NIH, I was doing work that was what I thought was fairly revolutionary. I was really gifted to be able to work with people that from different fields that were really quite talented. I did a lot of work with sequencing with our genomics group at the NIH, and I did a lot of work with mass spectrometry with our clinical chemists. And one of the early projects that I started working on was identification of bacteria with mass spectrometry. And although it wasn't the first mass spec tool that I used, we eventually migrated to using MALDI-TOF. And this was now about 16 years ago when we were doing this work. After 4 or 5 years, I became convinced that the technology was reproducible and, all right, thank you. That should sound a little bit better, and I won't lose my voice quite as quickly. We started working with mass spectrometry, and I was partnering with Bruker in the work that we were doing. The original mass spectrometer that we used was probably about 6 feet long, and it had to be put horizontal on the floor rather than upright because it was actually too big. They've obviously made a lot of changes in the mass spec systems that we use today, but the thing was that it was really dramatic, the work that we were able to do with the system. But when I went to try to publish it, the response I would get from the clinical journals was that this was a tool that was really a research tool. It wouldn't be used in the clinical laboratory, and I wasn't able to publish it in at least the American journals. I published in the European journals. And I look back now and have to laugh at that, because if you look in most laboratories, they either have a mass spectrometer system or they're considering one. It really has truly revolutionized the way we do identification of organisms. And I would predict, and since I'm in a predicting mood today, I would predict that if we did a survey of all the people that would attend a meeting like this in 10 years, virtually everybody would be using mass spectrometry in their laboratory to identify organisms. I think it's fortunate that biochemical testing is going to disappear, even though I've spent much of my life writing books and giving advice on how to identify organisms biochemically. It turns out that it's not that accurate. And that actually was my fear when I got to the NIH and I decided I would start a sequencing program, because I thought that's the way we would identify organisms. And mass spectrometry really was a secondary consideration, but it turns out to be faster and cheaper and much more accurate. So let me start with a discussion there. The faster part is you can pick a colony, you put it on a target plate, you lyse the colony with formic acid, put a matrix on the sample, and within a minute you have the organism identified. That's about as simple a technology as you can imagine, and it certainly is very fast. I give 2 examples of systems that are in the market today. There are a few others, but these are the most common ones that you see, the BD Bruker Biotyper and the bioMérieux Vitek MS. Those are 2 systems that we see today. Both are highly accurate in identifying organisms. There are distinct advantages with one system versus another system. I'm not going to spend very much time talking about that. I really wanted to look at the overview of using mass spectrometry for identification. What I can tell you is that using MALDI-TOF, it will replace all of the biochemical and morphologic identification tests that you perform. And this means that you're going to have less consumables, less quality control tests, certainly less repeat testing that you would get with biochemical tests, and the need to do supplementary testing, all of which is going to reduce the cost of doing organism identification. The analytical sensitivity is actually equivalent to sequencing hundreds of genes, so the accuracy approaches virtually 100%. The reason I say it's equivalent to sequencing genes is because what you're going to do is see a series of protein peaks, basic protein peaks, that are extracted and detected using the technology. If, at least with the Bruker system, you can see this, if you have a single amino acid change, the molecular weight of that protein peak will shift in a detectable way. So for each amino acid change in your protein, you can identify that change. And because of that then, and because you're looking at literally hundreds of genes, you're sequencing hundreds of genes. And that's why this is so sensitive and accurate. And you can identify organisms like anaerobes just as easy as an aerobic organism. And in fact, when we adopted this technology, we made a number of changes in the laboratory. One included eliminating our anaerobe laboratory. There wasn't a need for it anymore. Up until that time, you did an anaerobic culture, and whether you had gas-packed jars or you had an anaerobic chamber, when you saw growth in the anaerobic plates, you first had to do subcultures to prove it was an anaerobe. You did some preliminary biochemical tests, and you did your definitive biochemical test, most of which required days before you had results. With multi-mass spectrometry, you look at your anaerobic plate, you see a colony, you pick the colony and you can identify it. You don't have to prove it's an anaerobe because you're going to get the name of the organism. So it turns out to be a much better workflow than what we've seen historically, and that was just one example. We developed databases for literally every major group of organisms, including the anaerobes, including molds, including mycobacteria. For the molds and mycobacteria, we used a different extraction procedure in order to release the basic proteins that you were looking at. But the essence of that was that for the molds, we were able to identify a mold within an hour of seeing it initial growth on a plate. The mycobacteria, it was the same thing. So it totally changed the way we did microbiology in those two areas of the lab, because now we had definitive identification and didn't require a skilled technologist to recognize the structural features of a mold or take weeks before you had a mycobacterial identification. And then finally, we were able to do tests directly from positive blood cultures, and we would have the identification of the organism in less than an hour from when it was first seen in a positive blood culture. So again, it totally transformed the way we were doing microbiology in the clinical laboratory. And that's why I would think back about the comments that this is a research tool and wouldn't be used in a microbiology lab seems a bit ludicrous today. What I'd like to do is not show you a whole bunch of data, but I'm going to show you about 8 or 10 different publications. And just to illustrate the value of using MALDI-TOF directly with a positive blood culture. The first study was one of the first published in this area. This is by Perez and co-workers in 2012. And what they did is they used MALDI to perform a rapid identification, and they also performed a rapid antimicrobial susceptibility test. And what they found was that you were able to, with the earlier reports, be able to get optimum therapy for their patient, that is removing from empiric to directed therapy, do that much earlier, which resulted in decreased length of stay and decreased hospital costs for their population. A second study that was also done the same year was able to show that the time for species identification using MALDI-TOF was reduced by more than a day, and there was 11.3% increase in appropriate antibiotics being used within 24 hours of that positive blood culture. So again, moving from empiric to directed therapy using MALDI-TOF. That's even before the antimicrobial susceptibility results were available. The third study performed in 2016 found the time for species identification and time to antimicrobial susceptibility results was reduced by 17.5 hours and 12.3 hours, respectively. And that meant that the time to optimum therapy was reduced dramatically, in this case by 19 hours. And then the French study, also published in 2016, found that direct identification of positive blood cultures resulted in clinical benefit for 24% of their septic patients. So what we see is that earlier identification, in many cases just the identification, improved therapy for the patients, moving from empiric to directed therapy, and also allowed them to have the patient's outcome improve, getting the patient out of the hospital and decreasing the cost to the hospitals. 3 other studies related to this. The first was by Patel, published this year, and they looked at directed MALDI identification of organisms, and they combined that with an antimicrobial stewardship program, and they saw they had reduced mortality, length of stay, and total hospital cost using this system. A second study in 2017, again using MALDI and the stewardship program, when they compared that with just MALDI alone, they saw that there was an improvement in therapy, length of therapy, and time to microbial clearance and hospital length of stay. So although we see benefits alone with rapid identification with MALDI, when you combine that with an antimicrobial stewardship program, you see even more significant improvements. And then the last study that I was going to cite is that MALDI reduced time to identification, initiation of appropriate therapy, duration of therapy, hospital length of stay, and mortality. Very convincing data. I don't go through all the specifics here, but as you look at the publications, very convincing data about the value of rapid identification, rapid susceptibility testing, and an antimicrobial stewardship program. So let me shift gears now. I just wanted to highlight the value of mass spectrometry for organism identification. Now I'd like to talk about antimicrobial resistance. And if you look at what's been projected to occur, we know that we have multi-drug resistant organisms that have spread globally, and we know that these organisms are associated with a relatively high mortality rate. If we don't make any significant improvements between now and what's projected the year 2050, we're going to see roughly 10 million annual deaths related to these drug-resistant organisms, which is going to far surpass the projections for cancer, many other diseases like cholera, diabetes, diarrheal disease, and so forth. So we have a problem that we need to address. And it's a global problem. The map on the right-hand side shows the world's population. The size of the circles that are in each continent reflect the impact of deaths related to these antimicrobial resistant organisms. And if you look in the populations in Asia and in Africa is where they project to see the most significant impact of drug-resistant bacteria. So we do have a challenge that we obviously want to prevent this from occurring, and we need to sort of look and see, how we can do that. Just to illustrate that this is not a recent problem, the graph on the right shows drug resistance and its increasing rate for 3 groups of organisms. The green line represents the increase in methicillin-resistant Staph aureus. The blue line represents vancomycin-resistant enterococci, and the orange line represents ciprofloxacin-resistant gram-negative bacteria. If we looked at resistance to the carbapenems, the extended spectrum cephalosporins, and to colistin, we would see dramatic increases just like these over the last decade. When we look at the graph that's and the figure that's on the right-hand side, this is the development of new antibiotics. And what you see is a steady decrease since the early 1980s to today, where we virtually have no significantly new classes of antibiotics being developed. So that if we're thinking that we're going to have a new antibiotic that's going to save us from these multi-drug resistant organisms, I think that's a naive approach. I actually think that even if they came up with a whole new group of classes of antibiotics, the bacteria have demonstrated they're much smarter than the chemists, and they'll become resistant to those antibiotics just as well. So I think that we do have challenges. What we can control is what happens in our hospitals and how we prevent the spread of these organisms, and from the laboratory's perspective, how we're able to detect these resistant organisms more effectively than what we've been able to do in the past. Now I'm going to focus almost exclusively on gram-negative resistance for the rest of this presentation, and I'm going to look at primarily the beta-lactam antibiotics and colistin. So most of my comments are going to be more restricted. If you think about the beta-lactam antibiotics, one of the most common forms of resistance is related to enzymes that degrade the beta-lactam antibiotics, or beta-lactamases. If we look at an E. coli or a Klebsiella, they've had beta-lactamases for years and years and years, and that's part of the reason why the first-generation cephalosporins, why the penicillins like ampicillin were ineffective. In 1983, though, we saw a mutation, a point mutation in some of these very common enzymes, and all of a sudden we had what is referred to today as extended spectrum beta-lactamases, or ESBLs. These simple point mutations took an antibiotic that was ineffective against, say, a first-generation cephalosporin or a penicillin, and now made the organism resistant to all of the penicillins and all of the cephalosporins. That was in 1983, and it was in Klebsiella with SHV. A few years later, the TIM variants were seen in E. coli, and then in the early 1990s, we saw CTXM, and again, primarily in E. coli, but also found in other Enterobacteriaceae. The CTXM now is the most common one that's found, and it's found globally. And just to illustrate that, this is a map of the world, and again showing whether these ESBLs, specifically the CTXMs, are endemic, are seen sporadically, or where we don't have data. And the reason I show this map is just to make one point, and that is that I'll make 2 points. The CTXM ESBLs are found virtually in every country in the world. Where they look for, they will find them. The other point that I'll make is that there is a number of different strains. So it's not like a specific strain developed and you could deal with a specific strain. You develop a diagnostic test that's specific for that. You develop a therapeutic approach for managing those patients. The reality is there's many different strains, so your molecular test for diagnosis is not going to be terribly helpful. and therapy is going to be quite variable depending on the strains that you have. I'll also mention, and I'll come back to this point in a few minutes, that for some of these strains that are resistant to the penicillins, the cephalosporins, and so forth, our in vitro test, whether it's a Vitek or a Phoenix test or a microscan test, is going to look susceptible. And so you need alternative methods in order to be able to detect these ESBL strains. I'll come back to that in a few minutes. If we look at one other group, and this is the carbapenemases, the ones that now, the organisms that produce an enzyme to break down all of the carbapenems, as well as the cephalosporins and the penicillins, virtually all of the beta-lactams now are ineffective. We see, again, these are globally spread. There's a number of different enzymes that have been described. I'm going to show data for two of them. The first is KPC, and again, where you see the colored filled-in areas, it shows where those organisms have been demonstrated to be present. I think this figure is a little bit misleading because, as an example, Brazil in South America obviously has strains. We just don't have data when this slide was created. The same is true for Mexico, for most of Africa, for much of Asia. We just didn't have data. The real value of this slide is just show that this organism has spread globally. The same thing if we look at MDM. This stands for New Delhi metallo-beta-lactamase. And again, we see a global distribution of the organism. A very nice map was created in a recent publication where they looked at the various genes that are responsible for these carbapenemases. And in this case, they looked at the 6 most common ones that are found. And I'd just like to point out a couple of things. If you look at Latin America and you look at North America, the bars are almost completely orange. And the orange represents the KPC gene. That's a bit unusual when you compare it to the other countries in the world. Most other countries have multiple genes that are circulating in their bacterial population. And if we look at the Middle East, the third from the right, what we see is the most common is the OXA48, the green bar that's in that figure. We also see NDM, the red bar at the top, and KPC, the orange bar at the bottom. Those are the 3 most common genes encoding for carbapenemases that you see in your region. Now, that's not just important for epidemiologic reasons. Much like with some of the strains of ESBLs, some of these genes will give you an MIC that looks susceptible, but in fact is resistant because it's producing the enzyme. And where it's most commonly seen is with the group that's most commonly found here, the OXA48s, the green bars that we see here. So the test that you run in your laboratory, your routine test, is going to underestimate the problems that you're confronted with these carbapenem-resistant bacteria. I was asked to make a comment about respiratory infections, and so what I decided to do is actually look at some data that's going to be presented at next year's ECMID meeting from some work that's being done with one of the groups at Becton Dickinson, where they've done a 3-year survey of 78 U.S. hospitals. And they're looking at the carbapenem non-susceptible strains that were found in these patients. Now, by non-susceptible means they either are classified as intermediate or resistant. And for this study, they found there was almost 1,500 pulmonary infections. The overall mortality rate was 13.7%. That actually is fairly low. I was surprised by that. But there is some other interesting data that was found in this study. For these patients with these carbapenem-resistant pneumonias, the mean length of stay in the hospital was 20 days. The cost per case was over $54,000. And about 1 out of 5 patients had to be readmitted to the hospital after discharge because their condition worsened. If you look now, just focus on the infections that developed in the hospital and compare those with the infections that developed in the community with these drug-resistant organisms, what we found is with the hospital-acquired infections, there is a 2.3-fold increased risk of mortality. The mean length of stay went from 9.5 days to 23.3 days, and the cost per case went from $24,000 to almost $74,000. So getting an infection with one of these carbapenem-resistant organisms in the hospital, and in this case pneumonia, illustrates how devastating it can be, both for the patient but also the healthcare system. So our ability to detect these quickly, to isolate the patient and provide appropriate therapy becomes critically important. I'm going to step back just a little bit and sort of set the foundation for the work that I'll talk about afterwards. I'm going to start with a discussion of antimicrobial susceptibility testing. And for those in the lab, all this is going to be fairly rudimentary, but for everybody to be sort of at the same level as I walk through this, we'll start with antimicrobial susceptibility tests, and there's really 2 general forms that are used. We have antibiotic dilution test and antibiotic diffusion test. The dilution test means that you take a series of antibiotic concentrations and they're diluted either in broth or in agar. The antibiotic diffusion test means that you put the antibiotic concentration in a disc or in a strip, and that's applied to an agar plate, where you measure the ability of the antibiotic to inhibit the growth of the organisms that had been inoculated on the plate. Antibiotic dilution tests, specifically microbroth dilution tests, are the gold standard that all of the other technologies are compared with. And those other technologies include a variety of automated platforms. I give 3 examples here: the Phoenix system, the Vitex system, and the Microscan system. But we have to remember that those tests, when they're validated and approved by the regulatory agencies, are compared with the microbroth dilution test. That actually becomes important when I talk about colistin. We'll see in a little bit. The relationship between the results that are generated by the diffusion test, let's say by measuring zones of inhibited growth, and by a broth or agar dilution test measured by MICs, is they're related. There's an inverse linear relationship between the log of the MIC and the size of the zone of inhibition. Because they're related, the results are equally predictive of clinical outcome. So in reality, whether you do a diffusion test or a dilution test, their ability to predict how the patient is going to respond, for the most part, there are some exceptions, but for the most part, they should be equally predictive. And both of the results can be reported either qualitatively, where you say it's susceptible, intermediate, or resistant, or quantitatively, where you give an MIC value. In this figure, I show the linear relationship represented in the orange bar. I have then 2 sets of lines, red lines and green lines, showing that if you start with a red line at the zone of inhibited growth, it's around 10 or 11 millimeters. If you extend it up to that vertical slope, or the orange line, and extend it over to the MIC, you can actually predict what the MIC value is going to be based on the size of the zone of inhibition. What it allows you to do then is to characterize whether the organism is in a resistant category, a susceptible category, or in an intermediate category. All of this becomes important because the accuracy of your susceptibility test, when you test an organism against an antibiotic, is going to be influenced where that organism falls in relationship to that orange line. So if it's very susceptible, it's an easy test to perform. If it's very resistant, it's easy to perform. But if the organism is distributed near the interpretive breakpoints that are established by the various regulatory agencies, then the test is going to be difficult to perform in a reproducible fashion. And again, this becomes important for certain specific groups of organisms. If we look at the accuracy of the MIC and the DIS test, that accuracy is determined by the ability of the antibiotic to diffuse through agar. So I'll give an example of colistin, which doesn't diffuse in agar, so you can't do DIS diffusion for colistin. The ability of the organism to grow rapidly in air. So if it's a slow-growing organism or if it's an anaerobic organism, you can't perform DIS diffusion tests. And I say DIS diffusion, it also includes disc elution or gradient elution tests like an E-test. An E-test cannot be performed accurately for a slow-growing organism or an anaerobic organism. The number of antibiotic dilutions that are tested and the measurement of the zone of inhibition will also determine the accuracy of the test. If you test more dilutions, your test is going to be more accurate. If you measure more finely the zones of inhibition, that also is going to improve your accuracy of the testing. The distribution of the organism and antibiotic in relationship to the interpretive breakpoints, I've already said, is important. Well, some antibiotics, like the fluoroquinolones, the extended spectrum cephalosporins, the carbapenems, and colistin, are all distributed near the interpretive breakpoints. So if we look at our test and we say it's not very accurate for certain antibiotics, more times than not, it's going to be due to this, because they happen to be distributed near those interpretive breakpoints. And finally, the presence or absence of an intermediate category. The graph I showed you showed an intermediate category between resistant and susceptible. Well, for some antibiotics, and colistin is the best example, it doesn't have an intermediate category. And because I said that the organisms are distributed near the interpretive breakpoint, small shifts in your testing results could shift the organism from susceptible to resistant, or resistant to susceptible. And that's one of the reasons why we have a lot of problems in doing susceptibility tests with colistin. I'm going to complicate this a little bit more, and I apologize for that, but it's also to illustrate that technical factors can influence the value of our antimicrobial susceptibility test. I mentioned that the interpretive standards, those lines that separate the different categories of susceptibility, intermediate, or resistant, those lines are established by regulatory agencies. Three examples are CLSI in the United States, UCAST in Europe, and USCAS, a new agency in the United States. We would think that they establish the same interpretive breakpoints, but the reality is that they don't. And so an example I'm going to give is with ciprofloxacin. And if we look just at Pseudomonas, if we use the interpretive breakpoints that CLSI gives us, and what we have here, I should step back, is a very large number of organisms that were tested against, in this case, ciprofloxacin, and then what was applied were the interpretive breakpoints from CLSI, UCAST, and USCAS. So same organism, same antibiotics, just different interpretive breakpoints. For CLSI, 37.8% of the organisms were called resistant. For UCAS, 48% were called resistant. And for USCAS, almost 84% were called resistant. So you literally get different results depending on whose standards you use. Obviously, that's a serious problem in my mind. Let's complicate it even more. In the case of CLSI, they decided that the interpretive breakpoints that they were using for the carbapenems needed to be adjusted. And they did this after 2010. They did 2011. Look, just an imipenem and the two values that I circled for Proteus and Morganella. So these are a very large number of Proteus strains and Morganella strains that were tested against imipenem. And when they applied the 2010 criteria and compared it with 2012, you saw significant differences. In 2010, all of the Proteus and Morganellas were susceptible to imipenem. But with the new standards, only about 2/3 of the Proteus were susceptible, and less than 20% of the Morganellas were susceptible. So if you follow trends of resistance in your hospital, all of a sudden it would appear like you have a major problem with Proteus and Morganella, when the major problem is the way we do susceptibility tests. So I think it's important as we think of the whole concept of antimicrobial susceptibility test to understand what can influence this. Some of it is that the organism actually becomes resistant, and some of it is because of changes in the way the susceptibility results are performed. All right, so now I gave you all that background. I'm actually now going to talk about three specific examples. We'll talk about ESBLs just very briefly. We'll talk about carbapenemase producing organisms, and we'll talk about colistin resistant organisms. Now, as I already indicated in the early graph, the world graph I showed with ESBLs, there's a number of strains that exist for those ESBLs. We would say the same thing occurs with the carbapenemase producing organisms. There are a number of strains that also exist for those. And so you would think that, you would hope that your susceptibility reports would be uniform for each of those strains, but in fact it's not. And in some cases with the ESBLs, and actually more dramatically with the carbapenemase producing organisms, they may be resistant because they produce these enzymes, but they actually look susceptible based on the MIC data that we generate. And I'm going to give the example with carbapenemase positive Enterobacteriaceae. So all of these are considered resistant because they produce enzymes that break down the carbapenems, the cephalosporins, the penicillins, and so forth. And in this study, they did susceptibility tests against imipenem for a large number of different organisms that were producing different classes of carbapenemases. And what you see in the second column, I've highlighted it in yellow, are organisms that are considered susceptible based on the MICs that were generated. And if you look at the bottom at OXA48, 37% of the resistant organisms based on MICs were called susceptible. Imipenem, it was 20%. I mentioned these two because particularly the OXA48, that's the most common one in your region. So as you do your susceptibility test, you are underestimating the amount of resistance that exists in your hospital. And that, again, is a serious problem. Let me illustrate how bad this is. This is a study that looked at carbapenemase producing Klebsiellas. And in this series of studies, what we're looking at is outcome from treatment. Now, the first vertical bar on the far left, the A column, they looked at mortality that was associated when the patient was treated with no drug that was active against the organism. And 45% of the patients died. Maybe that's to be expected. These are virulent organisms. These are all Klebsiellas, by the way. They're virulent organisms that produce an enzyme that degrades antibiotics. So 45% died. If you used a single drug that was active against this organism, or this whole population of organisms, you still had almost 40% of your patients die. And so what was obvious was that you needed combination therapy to have any degree of success, and less than 30% of patients died when they used combination therapy. What was sort of interesting from this study was that if your combination therapy included a carbapenem, now again, remember the carbapenems now are resistant. So you had one susceptible antibiotic and one that was resistant. There's an error in this slide. C1 should be without carbapenem. You saw mortality that was above 30%. If you added a carbapenem, that's where you actually saw the best outcome for the patients. And so mortality in that case was down around 20%. So the ideal therapy, even though the carbapenems are supposed to be inactive, if you combine them with what is clearly an active drug, then you had better recovery for those patients. You had the optimum recovery of the patients. Now, going back to what I said, you won't recognize a certain proportion of your resistant organisms based on the MIC test. Let's think about that. If you have an organism that truly is resistant based on carbapenemase production, we know that we have to use combination therapy. But if you don't recognize that it's resistant, then you could use monotherapy, and that's where we see the worst outcome. So again, we need to be able to recognize these resistant organisms, and we need to be able to select the right therapy in order to manage these infections. Now, there's a number of alternative ways in which you can recognize that you have a carbapenem-resistant organism. I've talked about MICs, or you can talk about disc diffusion tests, and you understand what the limitations are there. So you need an alternative test to help be able to demonstrate that even though it's supposed to be susceptible, it truly is a carbapenemase-producing organism. The most obvious way is to screen the patient with a molecular test. They're very sensitive, they're very accurate, they're rapid, and unfortunately they're expensive. And so you could not routinely, or probably would not routinely, use a molecular test to screen for the presence of carbapenemase genes. You can do culture on selective media. That turns out to also be accurate, but it's slow. It takes a day to 2 days to get a result. That's one of the disadvantages that we see with that. That's for screening. If you actually isolate an organism in culture, and again, you perform, let's say you perform your Phoenix or your Vitek test or your microscan test, you may not recognize the resistant organisms. So again, you could test each one of those with a molecular test, but again, that's fairly expensive and very few labs would do that. You can use a manual or an automated susceptibility test to do that, and in some cases it works and in other cases it doesn't. So let me give an example of some of those confirmatory tests. I'm going to give 3 examples. The first is the modified Hodge test, which is the agar plate in the upper right side. It's very difficult to be able to see from your seats, but what you have is a lawn of bacteria with, I think it's an imipenem disc that's placed in the center of the plate. And you see a zone of inhibited growth around there. Where the red arrows are is your test organism that is a carbapenemase-producing organism. And because it produces carbapenemase, it inactivates the antibiotic that's diffusing from the disc, and what you see is enhanced growth along the line of bacteria as it approaches the disc. That test, unfortunately, though, is very difficult to read. and particularly for organisms that don't produce very much carbapenemase. And I think for many of you that have used the test, it's not a test that it really is very reliable in the clinical laboratory. Another test is the CarbaMP, which is the bottom left-hand side, and it's a test that's very simple in concept. You take an antibiotic like imipenem, you expose it to an organism that is producing a carbapenemase, the enzyme hydrolyzes the imipenem molecule, producing an acid byproduct, and that's then detected by a change in pH or a change in color with a pH indicator in a broth suspension. Again, if the organism produces a low concentration of carbapenemase, that color change is very difficult to detect, and again, not considered very reliable. The last test is a test that was recently developed and launched in the BD routine susceptibility panel. It's called CPO Detect. And a series of wells are devoted to various inhibitors that are able to detect not only the presence of carbapenemase, but also to classify if it's a class A, B, or D carbapenemase, the three classes of beta-lactamases where we see these carbapenemases. In the study that have evaluated this test, there are two studies that were reported. The first was with a very challenging set of organisms that produce quite rare forms of these carbapenemases. And the test was able to identify between 96 and 100% of the carbapenemase producing organisms. It also has the ability to classify the carbapenemases in one of the three classes. That becomes valuable because there are new antibiotics that are effective against all of the class A and most of the class D carbapenemases, but have no activity against the class B or the metallo-beta-lactamase carbapenemases. So you want to be able to say that first, your organism is a carbapenemase producing organism, and then secondly, would it be susceptible to these new antibiotics that are active against classes A and D? And this test does a very good job of identifying those. We also, as part of the clinical trial, looked at the data and we see even better performance, which probably is expected since these are not the challenging organisms that were used in the first study. And we see excellent detection of carbapenemase production as well as classification of the organisms. Again, just to emphasize, this is part of a routine panel, and much like tests to detect ESBL producers in the routine panel, this one does the same thing for the carbapenemases. So as we look at problems, at least for some of these, we see solutions, and that was the main point that I wanted to make. Now, unfortunately, that's not the case with colistin. Colistin, we have a problem and we don't see a solution yet. And let me just illustrate this. Broth microdilution is considered the gold standard for susceptibility testing. Rarely do laboratories perform broth microdilution tests. It has to be with a full dilution panel of antibiotics under very tightly controlled conditions. If you did diffusion or gradient test, again, because colistin doesn't migrate through the agar, those are invalid, so you can't do those types of tests. And the rapid automated tests like Phoenix and Vitek 2, as well as tests like Microscan and Sensititer, are associated with a high proportion of very major errors. That is, a resistant organism calling the antibiotic susceptible. When we look at major errors, that is susceptible organisms being called resistant, we don't see that effect. Let's see how to say this. For the Phoenix test, there was mixed data that was in the literature, and so I'm funding the study with an independent team to look at a global collection of these colistin-resistant organisms. What we've found that with the plasmid-mediated resistance, it's able to detect these organisms accurately, but with the chromosomal-mediated resistance, right now it is not accurate. And so we are working with this group of investigators to change the algorithms to see if, in fact, we can appropriately classify colistin-resistant organisms. But that's work in progress, and I really can't predict whether we're going to be successful there. bioMérieux with the Vitek 2 has the exact same problem. And so currently, both CLSI and UCAS recommend that the only test that can be performed with colistin testing is the broth microdilution test. I would say the one caveat to that, at least in our experience with Phoenix, is that you run the test. If it's resistant, if it says it's resistant, it's truly resistant. If it's said to be susceptible, then it has to be confirmed. And molecular test in this case does not work well. There's a number of reasons for these errors. The organisms are distributed near the interpretive breakpoint. There's a lack of an intermediate category. We have what's referred to as heteroresistance, where you have a population of resistant organisms, but only a small subset express that resistance. That's reminiscent of the problems we had with Staph aureus and oxacillin resistance. But in Staph aureus, with oxacillin resistance, we found that if we lower the incubation temperature, if we add salt to the medium, and if we extend incubation, we're able to detect those resistant organisms. We just don't know if we can do the same thing with colistin. It wouldn't be those changes, but hopefully we can identify some specific changes that will allow us to detect that. I'm going to skip that, and I've already said that. The last comment is just about some miscellaneous antimicrobial susceptibility tests. Just to point out that for Haemophilus, we have standardized tests for both disk diffusion and microbroth dilution tests. For the pathogenic Neisseria, gonorrhea, and Neisseria meningitidis, we have disk diffusion and microbroth dilution tests using different media formulations and interpretive criteria, but those do exist. For the anaerobes, there's really only one anaerobe where there's a standardized test. That's for Bacteroides fragilis group. They can be done by agar dilution or microbroth dilution using different media formulations. There is no reliable disk diffusion test or agar strip elution test for Bacteroides fragilis, as well as for the other anaerobic organisms. So anaerobic susceptibility test, other than in a research lab, is generally not performed and is technically difficult. So maybe some concluding comments, and I'm going to make these specific for the BD Phoenix M50, the new Phoenix panel or platform, and the Emerge panel. They offer significant advantages for the detection of resistant gram-negative rods. The Emerge panel consists of 136 wells, so you have a very large number of antibiotic and antibiotic concentrations that can be tested, as well as specific tests for ESBL production and carbapenemase production. What this allows us to do then is to do the confirmatory test directly at the same time that your routine susceptibilities are performed. And then finally, the second generation Phoenix M50, which was launched about a year ago, utilizes state-of-the-art design and expert system informatics, which I didn't really discuss in this presentation, but it allows us to generate highly accurate, reproducible antimicrobial susceptibility results. I think that's it.